Hello everyone, in this class we'll try to learn about apoptosis. Apoptosis is programmed cell death. Apoptosis is a Greek word meaning of falling off or dropping off, first described in 1972. It is a distinct process from necrosis. It is a form of coordinated and internally planned cell death which is of greater significance in variety of physiologic and pathologic conditions. Apoptosis is a programmed cell death that has genetic control. Genes of the own cells play important role in this programmed cell death. Apoptosis is a natural process and some of the examples are death of neurons in central nervous system during brain development, and synapse formation is an example of apoptosis. During fetal development, degeneration of many tissues like web in the fingers is the other example. Many blood cells like eosinophils undergo apoptosis. Mechanisms of apoptosis. There are variant stimuli which are going to initiate the apoptosis. Activation of cystine proteases in the cell called caspases trigger apoptosis. Normally, caspases are present in the inactive form in the cell and stimulated by external and internal stimuli, which are going to lead to intrinsic and extrinsic mechanism of apoptosis. Internal stimuli. The mitochondria release cytochrome and a protein called SMAC that causes activation of caspase 9, which induces apoptosis. In mitochondria, apoptosis inducing factor is located in the intermembrane space that migrates to nucleus and destroys DNA. External stimuli. External stimuli are various ligands that bind to the cell surface to activate apoptosis. One such factor is tumor necrosis factor TNF that activates enzyme caspase 8. Activation of caspase promote DNA fragmentation and chromatin condensation. Molecular mechanism of apoptosis. Initiation of apoptosis. Stimuli signaling programmed death act either on the cell membrane or intracellularly. Usually, it is triggered by absence of stimuli that are normally required for normal cell survival, such as absence of certain hormones, growth factors, cytokines, and activation of the receptors like receptors of tissue necrosis factor initiates the programmed cell death. Intracellular stimuli that may include heat, radiation, hypoxia, they initiate apoptosis, and also the genetically programmed events. Regulation of apoptosis. Once apoptosis is initiated, certain intracellular proteins provide signal for the final programmed cell death, which actually determine the outcome. These regulatory proteins may include the following. BCL2. BCL2 is a protein which is equivalent to CD9, the cell death gene found in nematodes. BCL2 is located in the outer mitochondrial membrane and control apoptotic process by binding to various cytosolic proteins. Usually it binds to BAX and BAD proteins that are known to promote apoptosis. It may also bind with BCL-XL protein that inhibits apoptosis. Another binding protein present in the cytosol is pro-apoptotic protease activating factor, that is APZAF1, which is a mammalian counterpart of gene CD4 of nematode. Thus, apoptote depends on binding of BCL2 with pro-apoptotic and anti-apoptotic proteins. Other apoptotic regulator proteins are the TP53, that is P15, P53 protein, caspases, BAX, and certain viruses such as adenovirus, papilloma virus, hepatitis B virus. They also help in the regulation of apoptosis. Steps of apoptosis. Apoptosis occurs in the following major steps. Initially, there will be activation of the FAS receptor, that is CD95 or FAS, the surface receptor present on cytotoxic T cell. When comes in contact with the target cell, they're activated. This leads to activation of caspases and subsequent proteolysis. The second step is ceramide generation, where the hydrolysis of sphingomyelin on the cell membrane generates ceramide, and ceramide causes mitochondrial injury. Then there will be DNA damage by various agents, and activated oxygen species like free oxygen radicals leads to apoptosis. DNA injury influences the nuclear protein p53 that induces the synthesis of cell death, promoting the protein BAX. Then occurs cell shrinkage, cell reduces in size and cytoplasmic condensation occurs. Then there will be formation of apoptotic bodies. Apoptotic bodies are formed and eventually they are phagocytosized, the dead apoptotic cells, and their fragments are identified by the macrophages, and they are engulfed by the phagocytosis. So this is the mechanism where the stimuli, whether it is external or internal, they're going to activate the fast receptor activation. There will be ceramide generation, which leads to mitochondrial injury and DNA damage, which helps in the formation of the nuclear protein, that is p53, and also they are going to induce the BAX proteins, and which leads to cytoplasmic and chromatin condensation, formation of apoptotic bodies, and later they will be phagocytosized by macrophages. So this diagram showing the extrinsic pathway and the intrinsic pathway. The intrinsic pathway is going to mainly the effect. It is going to lead to the mitochondrial injury, and the BAX is going to influence, it acts as a regulator, and ultimately the mitochondrial injury leads to release of cytochrome C that leads to formation of caspases 9. Whereas in extrinsic pathway, the Fas is going to bind, Fas ligands are going to bind to the Fas, and they are going to induce the formation of the caspase 8, and eventually caspase 8 is going to activate 9, so which forms the common pathway. So this is another diagram chart showing the intrinsic and extrinsic pathway, whereas the intrinsic is mainly by the internal toxic stimuli. It leads to inhibition of BCL2 or BCL-XL, which leads to activation of BAX or BAK, and which leads to damage to the mitochondrial membrane, releases the cytochrome C in the cytosol, which is going to cause a release of caspase 9. And extrinsic pathway, where the Fas ligand is going to bind to the Fas, that is going to eventually cause release of caspase 8, and that in turn causes release of caspases 6, 7, 8, which leads to apoptosis. Physiologic and pathological process. Apoptosis is a biological phenomenon which has both physiologic and pathological process. The physiological process is initiated at transmembranous or intracellular level. Then regulatory proteins like BCL2, they are going to regulate or organize the programmed cell death. Whereas some of the physiological processes occurs in 3 steps, as we mentioned, and they're influenced by many factors. And some of the example of physiological apoptosis are physiological involution of the cells in hormone-dependent tissues such as endometrial shedding in menstrual cycles, regression of lactating breast after cessation of breastfeeding, and normal shedding of intestinal epithelium, and involution of the thymus after childhood. Whereas pathological processes, they are initiated by the following factors like tumor cell death on exposure to chemotherapeutic agents, and transplant cell death by cytotoxic T cells that cause transplant rejection, and cell death induced by viral infection as occurs in viral hepatitis, pathologic atrophy following withdrawal of stimuli For example, when the prostatic atrophy occurs after orchidectomy, then cell death induced by radiation, hypoxia, and thermal injury, degenerative disease of central nervous system such as Alzheimer's disease and Parkinsonism are the example of pathologic apoptosis. Changes that occur during apoptosis. Pathophysiological changes in apoptosis confuses with the changes in necrosis. In necrosis, cytoplasm is homogeneously eosinophilic, and nuclear changes are pycnosis, that is condensation and clumping of the nucleus. There you see a karyolysis, that is the disintegration of the nucleus, and karyorexis, that is nuclear fragments into the small bits and disperses into the cytoplasm. In apoptosis, there is a condensation of nuclear chromatin and cell fragments into apoptotic bodies that are phagocytosized by the macrophages. The characteristic change in the apoptosis include that they may involve single cell or cluster of cell. The apoptotic cells become round or oval and reduce in size. The cytoplasm becomes intensely eosinophilic, containing condensed or fragmented nuclear chromatin material. Typically, inflammatory response around the apoptosis is absent. Though the cytoplasm is reduced, organelles remain almost normal. The cell membrane convolutes with the formation of membrane-bound spherical structures called apoptotic bodies that contain compacted organelles. The chromatic condensation occurs around the periphery of the nucleus, and phagocytosis of apoptotic bodies occurs by macrophages. This diagram shows the difference between the necrosis and apoptosis, where the necrosis is caused by the inflammatory action produced by the neutrophils, and they cause the external changes, whereas the apoptosis is the internal change which occurs and which leads to formation of apoptotic bodies later, which will be phagocytosized by the macrophage. Biochemical changes include proteolysis of the cytoskeleton proteins. Cross-linking of the protein molecules can occur. Fragmentation of nuclear chromatin by activation of nucleases. A glycoprotein molecule called thrombospondin and a phosphoprotein called as phosphatidylserine appears on the outer surface of the apoptotic bodies, which facilitate the recognition by macrophages for phagocytosis. Application: the identification of components of cell death control and effector mechanisms and the linkage of abnormalities in cell death to human disease, in particular cancers, has unraveled the research for preventing cell death. Understanding the concept of apoptosis has promising role in the future regenerative medicine. So this is in brief about apoptosis. Thank you.